Carrageenan is a material that can be obtained from red seaweeds, which is cultured and harvested in ocean farms in the Philippines, Indonesia, and Malaysia. This material is used as a binder in processed meat, providing a material texture and consistency for keeping its volume intact in storage. Its water retention properties keep the processed meat juicy and not dried or burned when cooked or baked. The property to gel at room temperature extend the applications in other products as a stabilizer and emulsifying agent in dairy products, beverages, and juices, and as a thickener for creams and sauces. To provide more information, let us hear from Dr. Romel G. Bacabag.
Three types of Carrageenan are commonly used by industry. Kappa, the rigid version of this material, Iota, the soft version, and lastly, the Lambda, which does not form a gel. In our previous study, mixtures of Kappa and Iota Carrageenan exhibited an effective averaging effect in the final properties of the end product. Carrageenan mixtures preserve the properties of the Iota and Kappa components, resulting into a viscoelastic hydrogel. Carrageenan mixtures have predictable properties that can be associated with the concentration of the Iota and Kappa components, where an increase in Kappa concentration makes the mixture more rigid. To exploit the natural biocompatibility of Carrageenan, we explore its potential in the fast-evolving 3D printing technology, where the gap between science fiction and reality is becoming slimmer. Here, the market for novel materials steadily grows, and we anticipate our participation to have significant relevance within the decade that foresees the presence of tabletop bioprinters in hospitals. We aim to develop a novel bioink using functionalized Carrageenan for 3D printing artificial bone tissue constructs by means of extrusion-based 3D printers. This type of biofabrication pushes the hydrogel ink through a narrow nozzle in order to manufacture on-site simple artificial tissues to more complex organs. The three types of extrusion technologies either use air pressure, contact force, or a screw mechanism to push the hydrogel ink. We will make use of the plunger-type extrusion by force contact in order to have better control and higher precision in printing pre-designed geometries. In this project, we will characterize Carrageenan hydrogel preparations, their deformability, and interaction with living cell inserts, using the methods we developed at the Medical Biophysics Group and the University of San Carlos. The response of functionalized Carrageenan hydrogels will be probed under extreme deformation conditions to mimic passage through the 3D printer nozzle. We will test hydrogel mixtures with living bone cells and optimize the resulting properties to 3D print the intended artificial bone tissue geometries. We will partner with the MCPI Corporation and the Holy Name University and have international academic collaborators. Daghang salamat.